Good morning, dear TY students. Today, we are going to discuss about the practical of the Ames test. To begin with, Ames test was given by the scientist Bruce Ames. Here, let us understand the principle of the Ames test. Ames test was developed by Bruce Ames in 1970s to test for determining if the chemical is mutagen or not. There are large number of chemicals. As you can see behind me, there are large varieties of the chemicals. But not all chemicals are harmful to us. Some chemicals may be useful to us, some may be harmful to us. But it is very essential to check whether chemical is a mutagenic agent or a carcinogenic agent or not. So to test this, he gave a test which is known as the Ames test. This test is based on the principle of reverse mutation or back mutation. Right? Reverse mutation, we all are knowing that mutation is any undesirable change which is occurring in the physical and chemical properties of the particular substance. Now, reverse mutation means in genetics we have discussed that any part of the gene or the genome is getting changed. Either some addition is there, either some deletion is there, or there is occurring certain amount of problems. Right? We have discussed various types of mutation. So if in the sequence any kind of mutation is there, then we can say that mutation has taken place. The agent which is able or which is responsible for causing mutation is known as a mutagen. The agent which causes cancer is termed as carcinogen. So this test is also known as bacterial reverse mutation assay. Test organism. Ames test uses several strains of bacteria. Here, particularly, we are using Salmonella typhimurium. Also, we can use various other strains of Salmonella and E. coli. But Salmonella typhimurium is the standard one to carry out the mutation in the gene that encodes histidine. So it is an auxotrophic mutant which loses the ability to synthesize histidine. Histidine, we all are knowing, is a type of amino acid utilizing the ingredients of the culture media. Those strains are known as histidine negative and require histidine for their growth. Culturing histidine, how we can culture Salmonella which is not having histidine in its composition is in a media containing certain chemical causes mutation in histidine encoding genes such that they regain the ability to synthesize the histidine. So if we are providing in the plate certain chemicals which make bacteria to produce histidine, we can say that mutation has occurred, but reverse mutation is taking place so that mutation is the chemical which is actually a mutagen. This Ames test is used to test mutagenic ability for variety of the chemicals. Now, what is the procedure of the Ames test? Let us understand that. In the test tube, you have to take the specific strain of Salmonella typhimurium, which is not able to produce the histidine. Then you have to check by for control plate, you have to check that it is not growing in the medium which is not containing the histidine. The salmonella is added to the plate with medium containing a small amount of histidine along with the mutagen which is to be tasted. Then we have to keep the plates for incubation for 12 hours. Then you will see that many colony produced by the salmonella that is mutated to restore histidine gene. Because we have added only a small amount of histidine, so the strains which will not grow without histidine, if they are growing, that means the chemical which we have added is able to synthesize or able to cause the mutation which enables the organism to produce histidine. Then when we have taken the salmonella which is added to the control plate with the medium containing a small amount of histidine, after 12 hours you will see that only few colonies result from a natural back mutation. What is back mutation? That only few organisms will grow because reverse mutation has taken place. What is the exact procedure? Let us understand that, so you will get a more clear idea about the Ames test. First we have to isolate the oxotrophic strain, then we have to prepare a test suspension of histidine negative Salmonella typhimurium in a plain buffer with the test chemical. Also add small amount of histidine. Small amount of histidine is required for the initial growth. In SEM 1 and 2 we have studied about the growth curve. We know that very for the initiation process we require the original compound. So a small amount of histidine is required for the initial growth of bacteria. Once histidine is depleted means it is lost, it is complete. Only those bacteria mutated to gain the ability to synthesize histidine form a colony. Means if you find colony in the plates, you will be able to say that the bacteria which have been undergone the process of back mutation, only those bacteria will be growing in the plate, or they will be only giving the colonies. Next, also prepare a control suspension of histidine negative Salmonella typhimurium, but without the test chemicals. Naturally, we are knowing that whenever we are keeping control, we are not adding the main ingredient. So here you are adding histidine negative Salmonella typhimurium without the test chemicals, and then you have to incubate it for 12 hours. Next, prepare the two agar plates and spread the suspension on the agar plate. Incubate the plates at 37 degrees for either 12 hours, 24 hours, or 48 hours, depending upon the growth. After 48 hours, count the number of the colonies in the each plate. The mutagenicity of the chemicals is proportional to the number of colonies observed. So here it is directly proportional. How much chemical is mutagen means how much it is able to cause the mutation, that will be directly proportional to the colonies which we have observed in the plates after 48 hours. If large number of colonies on the test plate is observed in comparison to the control, then such chemical is said to be mutagen. Naturally, if you have added a chemical in a particular quantity, particular portion, and you observe large number of colonies after 12, 24, or 48 hours, you can say that the chemical which you have added along with the histidine is a potential mutagen, because it has created the mutation, it has done the back mutation which has led the organisms to grow even in the absence of the histidine. Very few number of colonies can be seen on the plates, right? On plates, you can observe only few colonies. This may be due to spontaneous point mutation. Now, what is spontaneous point mutation? We have already understood that at a time mutation would have taken place on the histidine encoding genes. The suspension at 37 degree, the suspension is kept at 37 degrees Celsius for 20 minutes, right? So what we can interpret, suppose you are taking some chemical which is either mutagenic or carcinogenic, we are adding in the plate containing small amount of histidine, and we are taking the test organisms, that is Salmonella typhimurium, right? And after some time of incubation, if you find large number of colonies, that means you can say that the chemical is highly mutagenic, that means chemical is able to cause the mutation. Now, what are the applications of the Ames test? We all are aware of that practical application of the Ames test is to screen the chemical mutagen that causes mutation and are carcinogenic to humans and animals. Some of the chemicals used as a food additive, flavoring agents are also mutagenic as well as carcinogenic. Isoniazid is an anti-TB, TB that is tuberculosis drug, and it is also mutagen. So it can lead to the mutagen if large number of consumption of the isoniazid is done. Ames test adopted to use eukaryotic cell culture, yeast cell, as well as the animal model to test mutagens. Since Salmonella is not the best test organism to test the mutation for human beings, certain chemicals initially are not mutagen, but when they are entered into the body of any particular organism, or especially human beings, then they react with some other compounds which is present in the body, and they form a potential mutagenic agent. Instance can detect suitable mutants in large population of bacteria with high sensitivity. It is the test for mutagenicity and not carcinogenicity. Means if you want to prove that any chemical is carcinogen, you cannot use the Ames test, or you cannot rely on the results of the Ames test. Yes, you can of course perform, but the final result or final conclusion you cannot give based upon this test. However, most of the mutagens can be detected. If mutation is taking place, if the agent is mutagenic, then definitely you can use this Ames test. It is a bacterial reverse mutation assay, so the defective genes of the bacteria can be mutated into the functional gene. So it is also beneficial. Why? Because it is the reversion mutation assay. So a defective gene can be transformed into the functional gene. Defective means it is not giving any useful work, can be transferred or transformed into the gene which is having actually some function. I hope you all are clear with this practical particular, right? As we are having various limitations, we are not allowed to perform the Ames test in the laboratory because we cannot deal with the mutagenic agent. There are risks to various people, right? So I hope you all are clear and you got the basic idea how the Ames test gets to work, right? If you are still having any doubts, any queries, you can always contact me. Thank you so much.